Okay, my friends, another win for the good guys. You know that I've been talking about bacteria, and we're trying to get a database going of bacteria that's in the human body and all that. And the function of bacteria is to create enzymes. They are the factories that create the enzymes in the body. Well, this just came out this morning. It says, unheard of in structural biology, new research unveils enzymatic keys to immune system regulation. The enzymes are the keys to chemistry, because you cannot make the chemistry work fast enough without the catalyst. And enzymes are the catalyst. So let's go through this and see what they have to say about it. But they have apparently been able to, like, maybe 3D sort of structure these enzymes, and they are exotic. So I could see where it would be the first time to be able to structure them. But what they really have to understand is their role in the nucleophilic substitution process that creates life. And it's created by completely nibbling away at molecules instantly, and I mean instantly, creating all kinds of different half-lives and splitting up these particles so that we can use them. All right, the enzymes are the key to life. If you don't have the enzymes, you don't have the chemistry. And in order to have the enzyme, you have to have the bacteria, because the bacteria is the source of the enzyme. So let's see if we can make heads or tails of this. Okay, my outstanding friends, before we even start, you know that I do research, and that's all I do. I do not recommend any medical advice whatsoever. However, right now we're trying to put together a database based on the fact that all the research indicates it's microbiome issues that cause virtually almost all diseases, it appears. Now, what is causing these microbiome issues, and what are these issues, and is there any resolution to these issues? That's what we want to find out, and this is all research. So I'm not saying take this, don't take that. I say go to your doctor, listen to your doctor, do what your doctor says, and in the meantime, let's see if doctors can look into these new ideas, because sometimes these ideas come out very slowly. And we're trying to push this envelope forward as quickly as we can, because the research really does indicate microbiome issues cause all kinds of problems. And they do, because the microbiome is where bacteria live. Bacteria create enzymes. Enzymes do the chemistry of your body, and without the enzymes, you can't do that chemistry. An enzyme speeds up a reaction millions of times. So when that thing does its nucleophilic invasion, it has to have all these chemicals to break down all of the things that it's invading. And they break them down, they're gone. And it comes in and it replaces it with the stuff you need to make your body function. It's called a nucleophilic substitution and invasion. But in order for that nucleophilic invasion to happen, you have to have a ton of enzymes, because the enzymes break down all of these molecules that are in front of these invaders, and they wipe them out, and they go right in and they take over. So without the enzymes, you can't get the things to break down. An enzyme is an instantaneous breaker of things. Like I say, in a quarter of a second, it does more chemistry than that same reaction. If it was by itself, it would happen, but it would take years. All right, you can't wait years to get a little more nutrition or a little more oxygen or a little more whatever it is. You have to have that instantaneously. All right, so that's what the research indicates, is the enzyme is being the catalyst. Without it, you're not going to get that chemistry. The enzyme, you're not going to have the enzyme if you don't have the bacteria. If you've ever had any really serious problems with your gut, and you don't have the bacteria to break things down, you're going to have gut issues. And then those pass on other issues. That means you don't have the chemistry in your body to maintain your membranes, to do all the things you need. And primarily it's membranes, because most all diseases are invasion of membranes. So this gets very deep. But what we want to do is isolate what bacteria is in what people and what issues those people have compared to the bacteria. All right, quantities, species, and the conditions, because there's a lot of stuff to consider here. What do the people eat normally? Or what kind of diet do they have? What kind of water are they drinking? All right, what's the air they're breathing in there? What is their environmental issues where they work and so forth? All of these things play a part. And now they are realizing that without the right chemistry in your body to deal with these things that you come up against, you're going to be ill. And these chronic long conditions, they claim now, as this is talking about, it's unambiguous biological. And biological, they're talking about a whole series of things, but one of them is the gut microbiome. And that was something that was consistent across the board. It appears that virtually all these people that had these issues had a whole different set of microbiomes than the people that were healthy and had no chronic fatigue. So let's dig into this a little closer. But again, this is research only. There's absolutely no, I'm not telling you to take anything, don't take anything, but we need to pursue this research. And in order to do that, we need a database. In order to get a database, we need a study. In order to get a study, we need somebody like my friend Andy Marino, who is a bacterial engineer, triple PhD. He owns a business, and they go out and set up labs in food processing places where they have to make sure that there's not contamination. So this is a guy who knows what he's doing. And I've been studying this stuff pretty well myself. And we have Marguerite, who went to Harvard for microbiome and ribosomes and all that business, which is all this interplay between molecules to break them down to become resources to make your life work and give you vitality, give you strength and energy, clarity, even your mood. All of that stuff is a product of chemistry. Everything that happens to you, 100% product of chemistry. So let's think about the chemistry, and the only way you get the chemistry is the enzymes. The only way you get the enzymes is with the bacteria. So I think we should start right from who has what bacteria. Then we can expand it from there. But if we don't know, like this study did this exact same thing. They studied a whole broad range. Well, let's get into the study. Okay, so we are seeing this. This is brand new research, and this is strictly research. There's no medical advice given here whatsoever. We're trying to understand what bacteria is involved in what enzymes, and what enzymes create what kind of immunities, and that's exactly what they're talking about. New research unveils enzymes are the keys to the immune system regulation. All right, enzymatic keys. Now, that's what we've been studying right along, and we need a database of the bacteria, because the bacteria create the enzyme. And what is an enzyme, and what does it do? Enzymes are created by bacteria, let chemical reactions in the body happen millions of times faster than without the enzyme. The process would eventually happen, but it takes years, and in us it takes a quarter of a second. Because enzymes are not part of the product, they can be reused again and again. Once these enzymes are created, they click chemistry. They go click, 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 click. And every time they click, it's like shooting out a pulse of energy or something that destroys the other invader or breaks down some molecules into their half-lives. All right? Because all of these atoms have a bazillion different isotopes, which have half-lives. But we see them very not a whole lot, because we're not seeing them as they're getting collapsed millions of times a second. We see a whole batch of different isotopes, but in the body, they can take all these things and just reconfigure them. It's called nucleosynthesis. The nucleuses can be resynthesized to become new, different types of metals and minerals and everything. And it's very simple to understand that, because that's what happens with the daughter isotopes. If you look at, well, let's just look at it real quick, and then I'm going to go into the research that's being done now on this specific area. And I was thrilled to see this come out this morning. Absolutely thrilled. Unheard of in structural biology, new research unveils enzymatic keys to the immune system regulation. It's exactly what I don't disagree with that whatsoever. Okay, in a new study recently published in the journal Structure, Scripps research scientists present the previously undescribed structure of two of these nucleic acid degrading enzymes. These are enzymes. So they looking for the structure of them. How do these things actually work? So they have actually come up with the structure of these enzymes, which is PDL3 and 4. Now, understanding these enzyme structures and the molecular details is an important step towards designing therapies for the various diseases that arise when they malfunction. So when these enzymes malfunction, they're not working correctly for whatever reason, they want to be able to intervene with some new enzymes. And these are the different diseases that these enzymes can affect, is lupus, erythemosis, rheumatoid arthritis, Alzheimer's disease, and literally all diseases, I believe, have a root in the fact that there's no enzyme there to do the chemistry, to create the product, to intervene quickly enough. And that's because more than likely the bacteria is not sufficient to provide that enzyme. And again, the bacteria is the factory that creates the enzyme. No bacteria, no enzyme. No enzyme, no chemistry. And that's what they're looking at now, is this new research unveils the keys to the immune system regulation is enzymes. All right, I think I showed you this, but remember this: an enzyme creates chemical reactions that happen millions of times faster than if you didn't have the enzyme. Millions. So we got chemical reactions going on like crazy. Well, what are the reactions doing? Okay, we're going to look at isotopes of phosphorus. But there's isotopes, every one of these has a bazillion different isotopes. Now, we only know of a few because we can only, they're so fast when they happen in the speed of life. It's literally the speed of light. They happen millions of times faster. Now, what is the process that happens with isotopes, and when these enzymes work on them to make them just like that? Well, these are all phosphorus. Phosphorus is not just phosphorus. All right? It's all these different weights of phosphorus until you get to stable is in the middle. Right? Below that, it wants more particles to become up to stable. Above that, it wants to get rid of some particles to go down to stable. But there's all these different variations between every single one of these. There's a bazillion. I mean, there's literally thousands and thousands of these particles. We only know of a few because we can only see a few. But what is happening here? If this was phosphorus in the beginning, okay, it's phosphorus. It's phosphorus is phosphorus. But when it's done with its decaying mode, its half-life mode, it can be silicon, aluminum, magnesium. Up here it can be sulfur. So in other words, what I'm saying is this is what's called nucleosynthesis. It's synthesizing these other different atomic molecules, elements, by using enzymes. The enzyme goes in and nibbles off a whole bunch of things and does whatever it does with them, and the remaining part is the new daughter isotope. The enzymes are the key to everything, and they can't explain all these enzymes with the current standard model of atomic physics. It doesn't work. Here's the one that does work right here. Is dipole electron flood theory. With this, we have 1,823 particles make up a hydrogen. Helium is not, which normally is one big proton. Well, a proton is 1,823 dipoles. Right? Helium is supposed to be two protons, 2 neutrons. That's like almost 8,000 particles. It's not 4. And that's why when these things get working on them, all these enzymes, they tear them to shreds and put them back together. And that's exactly what an enzyme does. It either breaks something or it builds something. Before it builds it, though, it might just add on to things. I don't know. But an enzyme is a destroyer. All right? And a protein is a builder, but they both come from a ribosome. All right, just to prove my point about no bacteria means no enzymes. Bacteria are living cells. I'm going to show you these in a second. And they float around your body alive, reproducing. They have the capability of consuming waste. So they eat things of different types, all kinds of different types, because you have all kinds of different types of bacteria in you. They can reproduce, so they just continually live in you, and actually producing enzymes. They're the enzymes. Better said, bacteria are the factories. Bacteria are the factories that produce enzymes. No factory, no enzyme. Simple as that. No enzyme, no chemistry. Bacteria make enzymes, which make chemicals. And those chemicals are exotic. All right, I've got a big, long version coming up in a second, but I just want to make sure you understand about these bacteria and enzymes. These are the bacteria. They have their own type of DNA, and that DNA says, I am going to make ribosomes, these little blue balls, and I'm going to make those because those are the things that are the enzymes. Originally, they were in a ball form. Now, these are single-celled bacteria, and there's thousands and thousands of them in your body, different species, and there's more of them. There's literally trillions and trillions and trillions of them in you, and there's more bacteria in us than there is us in us. They are ubiquitous in our bodies, and if they're not there and they're damaged, you do not get the chemistry, because that is the chemistry. Now here's what happens with that chemistry, the ribosome. It comes out, and there it is right there, and then it unsheaths. Now, I'm not sure when it unsheaths, but I'm pretty sure when it senses that it's up next to something that it wants to kill, it unsheaths itself and turns into an enzyme. Now, here's where I was talking about before, enzymes and proteins. All right, let's take this slow, because it does both the same things. And a virus is also a ribosome. That's why viruses aren't alive. They take over in your body. Once they get in there, they take over the mechanisms and start to reproduce. Now, the enzymes are very similar because they create an enzyme which breaks things. All right, that breaks things up, food, and kills things and all that. Well, they also could become, instead of an enzyme, a protein. And a protein has all this really fancy network going on in it, but it's a builder of things. Now, it also could be a virus. We think of an enzyme as a good thing, which it is. But an enzyme is also a virus, which is a bad thing because it works against our body. Some of the enzymes are the good bacterial enzymes. They work with our body. That's what they're talking about right now, this big breakthrough of this, enzymatic key to the immune system regulation. And it's in this fluid-filled highway where these enzymes and bacteria scoot around through your body. And it's called the interstitium. It's a new organ system that they say is a new, well, it is. It's completely saturates your whole body with all of these fluid-filled highways where these little creatures can get through there and deposit the enzymes. But if you don't have the bacteria, you're in trouble. And the bacteria is something that we want to get a database of. So I'm going to show you some research that we've been working on, and it's only research. There's no medical advice I will ever give anybody because I don't have any. All I have is the research we've been doing, talking about, trying to establish some way of understanding what we're seeing. And I see they are starting to understand how important these enzymes are. Without those, you're done. All right, and without the enzyme, bacteria, you don't get the enzymes. That, in my way of thinking, that's really where the root cause of invasion is. Because enzymes create very, very exotic chemistry. Did I show you what an enzyme looks like? Look at this. This is what enzymes look like. All right, when that thing unsheaths, you get all these strands of magnetic, basically data. All right, that is nothing more than a magnetic signature of an enzyme. And when it gets up next to something, all right, if he sees a bad guy over here, he comes over and he goes, click, and the guy dies. It's good, done. It just tears his shreds apart, apparently. And then it goes on to the next guy, click, 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 never gets used up until it's, I don't know how long they float around for. I mean, they must degrade at some point. But they're supposed to come up and snap, 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 and all of these guys die, and this guy continues on without being damaged, unused. Now, let me show you something about how the immune system does work, and it's quite simple. Again, the bacteria is the key to creating the enzyme. So let's just take it real quickly. We know about bacteria. We know they have its own DNA. We know it creates a ribosome. The ribosome is nothing more than a huge, gigantic chain of these polypeptides, protein chains. And there's 20 different proteins, and they can add up in all different ways, and they program messenger RNA. Messenger RNA goes up, and it injects itself into our own DNA, I'm sorry. And how does it do that? RNA polymerase. Well, what is RNA polymerase? What is that? It's an enzyme. And if you don't have that where you need it, it can't open this up. That's an enzyme, and it does all this chemistry, breaking everything up, say, oh, you get out of the way, get out of the way, you get out of the way, get out of the way, it's coming in, come on, come on, get out of the way, get out of the way, get out. And it's doing this stuff. Done, case is closed. Now you're immune to whatever that was that this bacteria was programmed to produce. So, in my mind, we need to be able to either get new bacteria, or we need to be able to reprogram bacteria. Can we reprogram? Can you take certain bacteria, and that's the only kind of bacteria that's in that culture, and then just let it live with some kind of biology and then come back 6 months later and it's still exactly the same bacteria? Or will it try to find something it has to fight off and change its bacterial structure, its DNA, to produce new types of ribosomes? And then it goes, and this time, the same thing happens, and when it injects it in it, now you're immune to something else. That's how immunity happens. These are the messenger RNA immune instructions into our DNA. And once it's in there, you should be pretty well immune to something. You see that? That right there is the RNA polymerase, which is an enzyme. It's like got a bazillion, these are all little magnetic intertwined dipole moments, which creates all kinds of a gigantic magnetic signature, which is just stunningly complex. And it has to be exactly that, or it doesn't work. All right? If something is damaged here, it can either work adversely or not at all. But if it's just the way it's supposed to be, and the bacteria was healthy that created it, and the ribosome was a healthy, strong ribosome, and when it interacted, it popped out just like this, it should have so many magnetic signatures here that it instantaneously, everything happens, and whatever the chemistry it had to work with, it's all over instantaneously. That's what a catalase is. And catalase is a catalyst. It's an enzyme. It's in your blood. It removes excess oxygen. All right, because that's what's called reactive oxygen species in your blood. If you have extra oxygens, and you will all the time, they have to be removed. The catalase removes them, which is an enzyme. This is an enzyme, and this is for breaking open your DNA so that you can become immune to things. But that's also got to be created by some kind of a bacteria or a series of bacteria that create that. But as far as I know, there's no enzyme that is not created by bacteria. Therefore, every enzyme is from a bacterial source. That's as far as I can understand it. Okay, my friends, this is all about research and chemistry and bacteria and all that stuff. It has nothing to do with any kind of medical advice. So if you're here for medical advice, you're not going to get it. All right, so let's get into the research that's being done and how it affects these enzymes and this fluid-filled highway, because a lot of this stuff has not been really fully investigated yet. And I think the first thing we really need is this bacterial database. That's what we've been working on. So we're hoping for the best on that. And I don't think that's a big ask. You know, a bacterial database. What's the normal bacterial species in human bodies? Just a database, just a playing field. What's pretty much normal? I mean, we know normal most things, but I don't think we know normal what the bacterial strains are. And if I think we did know a vast database of just collecting a little bit of poop, basically that's what we need. And then have some kind of a test that tests what bacteria is in there and what quantities. And a lot of that stuff can be done with light now, by picking up the signatures of the light pretty fast. I think they might be able to develop a test or have one right now. They just go blip, and it says, okay, we got so much of this mineral and this one and that one, and then you can put it together as a profile. And then we also have to know what the bacterial species are. All right, so we know what's in there. But first of all, we have to find out what is the species that are in guts. I don't think they really know at this moment how many different species there are of bacteria. They know there's a lot of them. But I'm not sure how deeply they've gone at categorizing them and developing a database. And that's what we need, is a database. And then we can see what that database reflects on the general population. All these people are sick with a certain disease, and they're missing these certain bacteria as well. That'd be a place to start. And if all of these people were, whatever, you're going to look for patterns. Let's just go with that. We're going to look for patterns on the type of species, the quantities, and the lifestyle. And also we want to find out demographically, did something change? Is there something wrong with the water in that area? Is there something wrong with the food? Are they doing something, there's going to be some patterns arise. That's what I do, is research. So it's nothing to do with telling you to take some pills or something. No, I don't do any of that stuff. All right, so let's get started. Okay, we all know that they say you have to have a certain diet in order to have a healthy body. And yes, I agree with that. However, that diet doesn't just turn into these products all by itself. They need enzymes and bacteria to convert those instantaneously with the enzymes into food products. Otherwise, you can't digest them, and then you get stomach issues, and you get chronic fatigue syndrome and all these other things if you can't digest. All right, digestion is the most important thing in your whole body. All right, so they realized this, and they did a study on what causes this chronic fatigue syndrome that everybody's been complaining about for years, and they say, you're just a crazy person, don't worry about it, everybody gets tired. Well, 8 years later, after the study started, the results of the study finally out in one of the most thorough investigations to date. Researchers took a deep dive into a small group of only 17 people who developed this chronic fatigue syndrome after an infection. All right, listen to this now. They got chronic fatigue syndrome after an infection, and they found distinct biological differences compared to 21 healthy controls. All right, so they got 17 people that had an infection, and they got 21 healthy controls. Distinct differences in biology. Well, what distinctly was different? All right, here's what they found. It says, overall, what we show is that this chronic fatigue syndrome is unambiguously biological. That means there's no question whatsoever. It's in the biology of your body, unambiguous. And it has multiple organ systems affected. Now, I don't know if that's in every person that has chronic fatigue syndrome, they have multiple systems, or is it one guy's got a liver problem, another guy's got a lung issue, another guy's got this, or is it all in one person when it's chronic fatigue? It could be either way. So let's see what they have to say now. Neurologist Avindra Nath, lead researcher of the study, clinical director, National Institute of Health, Neurological Disorder and Stroke, said in an interview with JAMA. So it's unambiguous. There's no question whatsoever. It is biological. It's causing this people to be fatigued. You have to digest your food, or you're not going to have that nutrition, and you're not going to have that energy source. And you have to digest your food using enzymes. That means you have to have the bacteria available. So that's where I'm coming from, is the microbiome. And so we need to know who's got what bacteria. So that's where my focus is. But let's go on to what they have to say. They say doctors dismissed this chronic fatigue as psychosomatic condition in all patients, and they're in their heads, and everybody gets tired, and don't worry about it. But now there is little doubt a host of biological changes underpin chronic fatigue, a host of different biological changes. So you can have all kinds of things, all kinds of different diseases, it appears, a host of different changes. What are those hosts of different changes? It's a systemic disease. Okay, my outstanding friends, this is going to be interesting. It's completely research just related to the new discoveries that have been made inside the human body, which is this fluid-filled highway that appears to carry all the enzyme, well, not all, but primarily is enzymes and bacteria live in this fluid-filled highway, and they can scoot around the whole body almost instantaneously. And this was not known until 2018 how sophisticated this system is. And because of that, the research into enzymes and bacteria and the chemistry that they produce and how they fight back against diseases and how they break down foods and how they make proteins to build up body tissues, and all of that stuff appears to happen primarily in this layer. And there's a lot of chemistry, there's a lot of all kinds of stuff, but it's just research. This is just research. I can't tell you to take anything, don't take anything. I can't tell you what'll make you healthy, won't make you healthy, but I can tell you this. If we don't know what bacteria lives in the human body, it's unlikely that we're going to ever be able to cure diseases, because bacteria are the factories that create the enzymes. And the enzymes are the only thing in your body that can work fast enough to do the chemistry of life. So, right, think about what I just said. Bacteria, which live in your body, and you have, and I have, thousands and thousands and thousands of species, different types. And every single one of them, almost, I can't say every one of them, but primarily their function is to create enzymes. Because they say they're the factories that create the enzymes. The enzymes do things in a quarter of a second that could take years and years and years if it wasn't for this catalyst with this enzyme. And it does it in a quarter of a second, at literally the speed of light. This is all magnetic charges, basically. And an enzyme is just a ball of magnetism, a bazillion different magnetic moments. And it's designed to hit chemistry and just instantaneously break it into billions of pieces. I mean, instantaneously. Like I said, I'm going to read you some of the text on how fast these things work and what they do, and I'll show you the actual molecules, an artist's rendition of it. But it's easy, once you understand. Well, you'll see. I think you'll be able to understand. It's not that big of a deal. It really isn't. But why I'm showing you Andy here is Andy Marino is my good friend, and we've been working 5-6 years together. And primarily, I've been asking him questions. He's a triple PhD bacterial engineer, and he knows all this stuff. And he owns this company, and they had a whole batch of labs all over the place where they're testing food for safety reasons. Very sophisticated lab, or labs. They go and they put them into your facility where you're processing your foods. That's my understanding of what he does. I really didn't get involved with what he does much, other than to just ask him a lot of questions about bacteria, and a very pleasant guy to talk to. Anyway, we now want to do the study on a database and create a database to track what kind of bacteria is in the human body. Now, Andy knows all this stuff. He's deep into this, and I understand it pretty well myself, very well, as a matter of fact. Not to brag. I ain't bragging if it's true, and it's true. Now, what we want to do is find out, is there any correlation between different strains of bacteria that's in the human body and different patterns of disease? All right? If you have a certain bacteria and you have a certain disease and nobody else has that bacteria and they ever get that disease, well, maybe that bacteria is causing you problems. Now, why do you have that bacteria and somebody else doesn't? That's the first question. So there's going to be, one is going to be about why you would have excess bacteria versus somebody who doesn't have that bacteria. Something is keeping it suppressed, because you're exposed to virtually every bacteria there is, almost, on a pretty regular basis, because, not every bacteria. I make these statements. Let me just put it this way. There's a lot of bad stuff that goes into you that never harms you. It doesn't, it just goes right through it. Everything's good, because it's pushed back at, basically. And that's basically within the membranes that surround all your organs. They have what's called phospholipid membranes, and there's a junction in there, and it should be a pretty tight junction in these membranes that keeps them all glued together so they can float around. But let me explain it when I show you the information on that and the anatomical shots and all that stuff, because the membrane is basically the most important part of you. And this is now just realizing that membrane also contains this fluid-filled highway, which was they never knew it before. Let me get started in here and show you this specifically. And this is is for doctors as well, because again, this is pretty new stuff. And this fluid-filled highway appears to be very, very heavily involved in your immune system. It's like your first line of defense. And then your bacteria is what are the defenders. So the bacteria becomes extremely important in the process because it is the factory that creates the enzymes. Once again, this is 100% research. And all I want to do is have Andy, set up database, get a database going. It's going to require some funding. You can't do something for nothing. But Andy's a good guy, a fair guy. He understands this business. We're trying to help kids. The kids have virtually no immune system when they're born, and things in the environment are getting very, very invasive to what we always consider to be immunities, which is just a natural immunity you get being exposed to things. But the things they're getting exposed to now is damaging the bacteria that's in our body. That's my opinion at this point, is the bacteria that's in our body creates the chemistry, which is the enzymes. And if you don't have them, you're done. That enzyme, if it's not there, you're not going to get the chemistry that enzyme was designed to provide for you. That was not just an accident that enzyme happened. That enzyme is there because a bacteria, for some reason, was created somewhere long ago, or even just recently, to say, hey, I see a problem over here, and here's my program for you. Make these little balls called ribosomes. So the bacteria says, I'm going to make these little balls called ribosomes. I'm going to squirt them out, and when they see a problem, they're going to pop out of their little shell, and that molecule is going to attack. And I mean attack. These things are instantaneously, like I said, in a quarter of a second, it will do what would take many years if it wasn't for the enzyme. So just think about that. Without them, we're done, and the only thing that makes them is bacteria. Okay, I do a lot in the subatomic realm, and my dipole electron flood theory solves all of this. Now, here's the problem. They don't understand isotopes. And there is no way to work with isotopes with the current model. This is all iron. All right, here's iron right here, Fe. All right, it's iron. It's just iron. Well, what is all this? These are isotopes of iron. They're not exactly iron. They're a little lighter than iron is supposed to be. You see this? These are the half-life. It can stay at that range, but only for a certain milliseconds and nanoseconds and so forth. But here's the isotopic mass. In other words, the isotope, how much does it weigh? All right. Basically, this one weighs 45, 46, a little more than 46, almost 47. Then we got 47, almost 48, 48, almost 49, 49, 50, 51. Then you get into some crazy electron volts, and then you get into some other stuff, which it's the same sort of thing, but it's in an energy level. Let's just go with that. This is where they say it's observationally stable. So here's what they're actually seeing. All right, observationally stable means you got a ball of particles this big, and if it's that big exactly, it's stable. Well, what happens if you take a few off? I take a few of them off, it goes down this way. It gets lighter and lighter because they're made up of all these tiny particles. They're not made up of big balls like that or to have a couple of quarks in them. You can't account for this with the standard model. It doesn't work. This does. So the more you take off, the lighter it gets going down this way. All right, and then when it decays, you see here. All right, we're talking about iron, Fe. Well, you got manganese right under it, and you got cobalt on the other side of it. And you see this? and chromium too. It can go down from here to manganese to chromium. All right, and then when you get above stability, it goes the other direction. See, stable, stable, stable. Now we come up to cobalt. It goes from here up to cobalt. See, they're all cobalt. But what happens is they're almost there. What I'm getting at is they're a big ball of electrons, and nobody knows this. This is not known. I'm not kidding you. They don't know this. They think the standard model doesn't work. They know that's true, but they don't realize that everything's made of dipoles. And this I've shown a mazillion times. Here it is right here. This is the dipole electron flood theory. And this is what the proton looks like. It's a black core with all tiny electrons everywhere, though, not just half a dozen or so on the outside. And this solves everything. Dipole electrons. It's just like every one of these is a dipole. It's the same thing as this. Only in the subatomic realm, the black part can go to the center, and the white part goes all the way around it. So you usually have a white and a black on a bar magnet. All right, and here's what happens. This is basically what the smallest particles there are looks like. We can break them apart, and we did, but normally they are attached tight, tight, tight. You can't pull them apart. That's what generating electricity is, ripping them apart and putting these in a little bucket so that you can use them. They don't weigh anything at all. They have no weight whatsoever. This has all the weight and has all the mass. Two of these they're nothing more than a bar magnet, a dipole. Two of them together make up a photon. That's light, and that is stable because this is back to back, and it has a big field surrounded it, and it says you stay out and I'll stay in, and that's fine. Here, it's got like just a half a field, and it'll say I'll attach to you, you attach to me, we're good to go. All right, this is not stable. That is stable, and then the next zone of stability is when you get to here, which is 1823, which is a proton. So if you're here, you're radioactive. You want another batch of the particles here. And what happens is you may come against somebody's DNA and go and suck off some of their particles and damage the DNA. Who knows what you could do. But radioactive means you're going to desperately want to interact with somebody else's particles. Radioactive. Okay, my friends, this is important to understand. This is nucleosynthesis. I'm always talking about nucleophilic substitution, nucleophilic substitution and invasion. This is exactly the same thing. This is the product of nucleophilic invasion. It's nucleosynthesis, where it's the process that creates new atomic nuclei. That's these things here. New products, new actual atoms from pre-existing nucleons, which is protons and neutrons, which are the atom's nucleus. So it's taking a nucleus and converting it into a new atom, which I just showed you, or maybe I did or I didn't, but I think I did. It's where the iron can convert up to cobalt or back down to manganese, depending upon its isotope value, and then how hot it gets, if it's in with acids, if it's in with salts. All of that makes a transition. And these are all transition phases creating new atomic nuclei. That is nucleophilic invasion, nucleosynthesis. All right, I've been pushing this over and over and over because people just don't understand it. And I don't think very many people understand it, even if they're working in the atomic realm, because they can't account for isotopes. There's just no way. If they're going to stick with the atomic model, you can't account for isotopes. It can't be done. All right, this is very, very simple chemistry. These are supposed to be the number of protons, and then they throw in a bunch of neutrons, but each one of these are protons make up atoms, and the more protons in a ball, the higher up on the periodic table you go, and they get heavier and heavier and heavier. Well, deprotonate means to remove a proton, which means change it from one atom to a different atom. They claim all of these metals and things were all produced in the Big Bang. Well, then they talk about deprotonating, which is to remove a proton. To remove a proton. So it makes it into no longer the same thing it was. Well, guess what that is? That's nucleophilic substitution. And when you depronate, this is the nucleophilic group as it attacks, it depronates. It changes it into a new molecule. And not only a new molecule, a new atom. So it's no longer iron, it goes down to manganese. Or it's no longer iron, it goes up to cobalt. What it does, it comes in and it's a nucleophilic attack. All right, so it breaks these particles apart. That's not really supposed to happen. I don't know if they understand what they're talking about here. It says, this is an example. A tackling nucleophile is an alcohol rather than an amine. That's why the enzyme, so you have to have the enzyme to do this job, is called a methyltransferase. In both cases, though, the basic amino acid side chain is positioned in the active site in just the right place to deprotonate, to break the proton off of the nucleophilic group as it attacks. All right, so now we're talking about nucleophilic invasion and substitution, which is how all my fossils were formed. And that's what this is about, leaving groups in biochemical reactions. Biological, chemical. Reactions. Okay, I'm going to make this very simple. Everything you have in you, or me, or any creature, they have membranes surrounding certain types of tissue. This is like skin, and this is like the floppy stuff underneath. They call it the interstitium, which is it's like a membrane. Now, there's a layer in there they never really realized before. They thought it was a dense, solid layer, but it ended up being a fluid-filled highway. And that's this right here. This is the new organ system that they realized, meet your interstitium, a newfound organ. That's that white layer right there. You see it? That layer runs everywhere in your body underneath a layer of skin or fascia, the coating of something. And this is a membrane. Now, I always knew the membrane was here. They just thought there was a thick wall, a mat that coated it, because by the time they did the autopsies, all the fluid had come out of it, because it's just fluid filled. It's just a big floppy thing of fluid. But what happens in that fluid is the bacteria and the enzymes run through your body, and it also picks up all the debris. It's basically, I believe it's more the lymph. But that needs to be, they just figured it out in 2018. Now, I have seen that in my fossils for many years, the interstitium. Now, what happens in that interstitium, and in blood in general, you have all of these metals, and they have charges on them. See the plus 3, 5, 2, 7, 6. All right, now, they make all these colors. All right, that's the transition metals. And those are in your blood. And when I say they're in your blood is just literally almost 100% metal, except there's a lot of water in it too. All right, here's the key. Enzymes, which you need to do these very complex chemical jobs. They're complex chemicals produced by bacteria. You don't have the bacteria, you don't get the enzyme. You don't have the enzyme, you cannot do this complex chemical stuff, which I'm going to show you in a second. Now, they cannot reproduce. They can only be manufactured by bacteria. Enzymes can only be manufactured by bacteria. And the enzymes don't actually consume waste. Bacteria does, though. Bacteria will go out and gobble up waste and deliver it. But they speed up chemical reactions without getting used up themselves. The enzymes go in, and it's called click chemistry. So the enzyme is going to be very, very elegant. Let me show you what an enzyme looks like, and I'll show you how it works in your body. All right? And they accelerate, listen to this, enzyme accelerates the rates of reactions by well over 1 million. So reactions that would take years in the absence of catalysts can occur in fractions of a second if they have the right catalyst. You got to have the right enzyme. The enzyme is the catalyst. The same word. Enzyme, catalyst, same. And what is it? There it is right there. It's like this, and every one of these little spots is a magnetic tip. And what it does is, as far as I can determine, these things float around in your body, and when they see an invader or they see a piece of chemistry that they want to break down, which they're designed to do, these are very well designed. They click, they go click, and it's gone. Click, gone. Click, gone. Click, 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 click. And they don't get used up. They just hit it with like a magnetic pulse, almost like radioactive, and it kills it. But it's designed just to kill that specific thing. That's why these things are so elegant, and they're created by bacteria. Now let me show you the evolution of this process. It's very, very interesting to me, anyway. And don't forget, you saw that elegant looking piece of chemistry that was the enzyme. Now glands, and you have them in all kinds of different organs. You have glands, but in the stomach they make acids and enzymes that break down food. Well, the enzymes are only made by bacteria. So your stomach is not really making an enzyme. The bacteria is making the enzyme. Enzymes are too complicated for just something to have happen. The bacteria have a program, and I'll show you that in a second. Now, these break down food in your stomach, okay? And your muscles grind it all up in there. Then you get to your pancreatic juices, and they make digestive juices, which is nothing more than more enzymes that have enzymes that break down more carbohydrates, fats, proteins. Now, if you don't have those enzymes, you don't break that down. They say, oh, I'm allergic to this. I can't do that. I can't eat this. I can't eat that. Well, why? Why not? You can't digest it. Well, why can't you digest it? Well, I don't know. But it appears that you have to have that enzyme to break it down. So what we want to do is a survey, a study, a big study. And this is all about that research. I'm not telling anybody to do anything, but we want to find out, is there any relationship to the bacteria that's in your, really in your feces? Because we want to do a very, very huge study to find out what you have in your bacterial profile. All right, what bacteria can we find? Now, there may be other bacteria that doesn't come out in the digestive system, but pretty much, I think most of it does. You can find, we could refine this, let me put it that way. But we need to know excessive amounts of this compared to that, excessive amounts of this compared to that, none of this, what happens? Too much of this, what happens? And make a database where all you got to do, it's got to be automated. It's going to take some money. It's going to take a little time probably, but I don't think too much to get a good idea. Is this a valid way to think? And I believe it is, because I believe if the bacteria is not there, the enzymes will not be there. And if the enzyme is not there, this is what will happen, and I'll show you right now. And it's not pretty. All right, so let's start from the beginning. We got the fluid-filled highway. Right below it, in these little pockets here, is where the membrane is, and there's bacteria living in there that create the enzymes. They squirt them out in there, and the bacteria run them around through the body, through this fluid-filled highway. All right, because they found out that if you get a tattoo over here, some of that stuff goes down and coats your organs and so forth. That's how we figured out that was a fluid-filled highway, because tattoo stuff was going through the whole body. A certain type of chemistry. I don't think it's anything, I'm not saying there's anything bad about it, but they were wondering how that happened. And it was strictly in the membranes, in the fascia. So anyway, and that goes back years and years ago. I was working with some other people, and they mentioned that the guy in Germany. So there's your fluid-filled highway. In it is these bacteria. All right. Now, bacteria has its own DNA. You see this? Its own DNA right there. All right. That DNA is just like us. Us, it says make four. Well, in my case, 5 fingers, 5 toes. 2 eyes and all that business, and 2 arms and everything. It's all of that stuff is in this. It's a program. Well, this says make these little balls, these ribosomes. See here, ribosome. And what's that ball all about? That ball is what holds on to the enzyme. And it's just like a pill. It squirts them out just like pills. And it says if you see something coming down here that has this certain magnetic signature, pop open and kill it. And this is okay, and they squirt them out. All right, so now this has its own flagellas. Most of them have flagellas, and they spin like crazy. And I think they can go very, very, very fast through your blood system. Because you can react very, very fast to any kind of an infection, a drug, so forth. Boom, just like that. It gets into your blood system. Now, so here you got shooting out these balls. Well, what do these balls look like when they come out? They have a sheath around them. All right? The sheath pops off, and out comes the enzyme or a protein. They're basically the same thing, only this breaks things, and that basically builds things. All right, so are they enzymes or are they proteins? Well, they're either or both. Depends on the circumstance, but that's the things that come out. Now, remember what they look like. All these little fancy looking things, this is what they look like. And these are serious. And if they're not exactly like that, they don't work. And if they get to a certain temperature, they get invaded by heat, they don't work. That's why when you get to a temperature, I think it's like 105 degrees, something like that, you're in danger of getting damaged because your enzymes shut down. A lot of this is because your enzymes won't work at these temperatures. High temperatures is too many extra electrons. You see, here's what happens to enzymes when they get exposed to heat. And it doesn't have to be high heat. It's not that high, let me put it that way. Enzymes are protein molecules, which I showed you. They're enzymes. They're really exotic enzymes. They call them protein molecules that get denatured at high temperature. Denatured means they don't work anymore. They're not a natural chemical anymore. So if that isn't like that, and it's like this, it doesn't work at all. So here's what happens. They say high heat breaks the hydrogen and ionic bonds, leading to disruption of the enzyme shape, just like I showed you. What happens is extra heat comes in here and it pops one of these off. And it goes like that, and it changes the shape. All right, disruption of the enzyme shape. High heat, hydrogen, and ionic, which means extra electrons. Ions are extra electrons to these bonds, leading to disruption in the shape. All right, so that's what destroys an enzyme, is heat. All right, so the enzyme loses its activity, no longer binds to the substrate. It doesn't really bind, it's just supposed to click. But if it doesn't have the right spots to click, it doesn't work. So it can't kill the thing it wants to kill. And that's at a certain temperature. Now, but again, if you don't have the enzyme, if you don't have the bacteria, you don't have the enzyme. That's the key. That's why we need to do this study. And again, 100% research. Because everybody's digestive system is going to be different. Andy and I were talking about this today. He brought out the point. He says, is there some profile that we could come up with for, optimal for everybody? And I said, well, that's a good point. I don't know. will certain types of bacteria stay in you and live in you and everything's fine, and then just one day all of a sudden you have a ton of fat, which you never eat fat? Or will that your body be able to react to that? Or will you have some gaseous reaction in your stomach because you can't digest it and you can't work with it? I don't know. You know, if you have like a consistent diet, is that good? At this point, I'm starting to think yes. And I'm starting to think that what the diet is ultra important. Everybody said, it was always said, you are what you eat, it is true. You become what you have eaten. And you need all of these different little byproducts to come down to service all of your tissues and everything. And that's all created by enzymes. Nothing in your body could work fast enough without these enzymes. That's chemistry. That I understand. And they understand that too. I showed you, I think I showed you, just so we don't lose each other. I showed you about the glands creating the enzymes and the acids. All right, so that's for your digestion. This is, in the blood. These are all the blood metals. We'll get into that later. It's almost 100% blood other than water. They're in aqueous solution. They're transition metals in aqueous solution. Let me just see. All right, here it is right here. Enzymes accelerate the reactions. These are enzymes by well over 1,000,000-fold. It would take years in the absence of these enzymes. But with them, they occur in fractions of seconds if you have the appropriate enzyme. All right, that's pretty clear to me, and I know that's a fact. And what it's doing, it's deprotonating, which is changing its, atomic structure. They say all these were made during the Big Bang. That's the only way you can make atoms. No, you can't. They say it right here: nucleophilic nucleosynthesis. Well, I had it up here before, but anyway. This is how your body creates the products it needs by, breaking down all the food that comes through your stomach. When they say breaking it down, it's breaking it down. And it's actually deprotonating certain things when you have these enzymes injected into wherever they're going. They literally change the atomic structure. All right, and they call it deprotonating. Well, what's the difference? You take the protons out, deprotonating. You just change the atomic nature. All right, and there's always these daughter isotopes. Isotopes, you can't account for them with the standard model. It can't be done. So we need to look at this in a different way and see if it makes sense with a database that collects data about these fecal samples. The first thing we have to do is come up with a method of collection which is easy and simple and non-invasive and so forth. And then, we need to know a database of what's the person's diet, first of all, medical conditions, any issues, whatever. And then see if it fits. And you may even find out that this whole region, because they're drinking a certain type of water, they have an issue with some type of bacteria, or they have too much bacteria because they're drinking that water. Something. Who knows where it will lead? This is research. This is trying to understand why some people get sick, other people don't get sick. They're right in the same place. They do the same things. They eat the same things. One gets sick and one doesn't. Why? Primarily, I would go for the digestive system. And, there is a certain poop chart that says if you poop in a certain way, you're probably going to be okay because your bacteria is dealing with all of the things that are coming through you. And they have to deal with all of them. Otherwise, you get gas and bloating and diarrhea and nausea and acid reflux. And, but that could be just from your diet, too. because you're not used to eating fatty foods or spicy foods or whatever it is, and you go out to do that just to have a good time, and all of a sudden, and people say that all the time. They say, oh man, I had this last night. Well, and a lot of times it's just having a good time, and you're not hurting yourself. But it would be nice to know if there was a real simple test you could do to see, is my stool sample looking good here? And then, if it's not, why? But this what we want to do is get to a database and maybe even get, like I say, a home sample. You sample yourself and just see, put it through a little, app on your phone. Click, am I good? So anyway, that's what we're trying to do. And like I say, these are all the different things that we're talking about. All right, the bacteria makes the enzymes or the proteins. They're both the same. They go out and they kill something in that separation zone. And once it gets through here into your good tissue, then you're in trouble. It's supposed to stop infections right here. And this is where you get your burning and your inflammation and all that in this area where something's trying to penetrate into your vital flesh down here. Out here, this is really like a glove around you. Your skin is like it's a coating to keep you protected. And it's expendable. But this, when you get down here, then you're in a whole different type of biology. So this is your protective layer, and this was never even known until just 2018. And they still don't understand, I don't think, the chemistry that's going on in there. So we know it's a fluid-filled highway, and these are the little things that are just zipping around in there, because you got to clean up all the debris as your body dies. Your body is dying every second right now. There's a lot of cells in you dying right this instant. Somebody's got to go clean them up. That's what these guys do. They go around and clean them up. They're macrophages, they call them. And some of them kill the bacteria that's coming in. I think that's still a macrophage. And other ones deliver the enzymes. And this is just sort of a geeky little thing to tell you. It's 20 amino acids. You spit them out in a peptide chain. It goes through the ribosome and it's programmed by the messenger RNA. Enough of that. All right, but here's the whole process. The bacteria creates the ribosome using these peptide chains. They get programmed using mRNA. It infuses into the DNA and becomes part of our immune system. But you have to get exposed to it first. And where does the bacteria come from that creates that ribosome? They must evolve along with infections. That's all I can tell you. Because you have to have the bacteria to make the enzymes, to make the mRNA, to make the change in your program so you're immune. I should explain to you how I discovered this in the first place. I discovered mud fossils. Prior to this discovery that I made back in about 2010 or so, 2012, I discovered bones and soft tissue could turn into stone. And it would just look like a stone, look like a mudstone. You say, oh, that's just a rock. Well, no, it's not just a rock. That was a bone. It's broken off here. There's still a taste of bone left in it. But there's a process called nucleophilic invasion and substitution. I think I just talked about it, where whatever this was made of, which in this case would be a lot of calcium, and then you're going to have inside marrow and all that stuff. Whatever a bone is made of, and it's the cartilage is up here, it got invaded and substituted. It's basically the same thing. It's called mudstone. In the center, it's going to be different because of the extensive amount of iron. Iron is the thing that protects and preserves. All right, let me just show you something about iron. All right, this goes back to 2013. Iron preserves and hides ancient tissues and fossils in fossilized remains. It preserves it. Okay, my friends, I'm going to wrap it up here, but as remember I showed you, these are all irons, and they're just bigger and bigger and bigger and bigger and bigger, or actually they're too small, and they're trying to get up to stability. And once they hit stability, and if you have more and more and more, it goes over stability. And then you give off these daughter isotopes. So they're breaking down these atoms to change from this atom to another atom from decay. It's from decay. And all it is you have too many little particles or not enough particles. And the only way to solve any of this is dipole electron flood theory. And there's going to be some differences to this in the standard model. There's going to be some differences to the periodic chart. The periodic chart's not right. Hydrogen is not one big proton like that and one little tiny negative particle that's out here somewhere. It's 1,825 or so of these dipoles, and they all push the black part to the center. They're all made of black and white, just like that. All right, and then these two together, they can come apart as I show. I'm sure I did, or I will. And then they're glued together, which this is normal state, and then the photons, and then the normal state of a proton is this, with all the black parts to go to the center. All right, I know I've been making some wild and crazy claims, but it's true. My old boss used to say it, if it's true, it ain't bragging, and it's true. We accelerated light. That's just light, not accelerated. That's accelerated light. The particles are right here that are accelerating. And these are what the particles are made of. This is what light photons are made out of. These are the exact identical same particles that Fermilab is looking for too. The exact same particles. And they do exactly the same thing. This one never changes, and that one turns into showers. So we've already seen these things. So I didn't want to have to show you this, but I'm not bragging. I'm just telling you this is true. This is the level that I'm on. I understand the subatomic realm, and this is what happens with this nucleophilic substitution. It is an instantaneous pew, and all of these different half-lives happen within an instant instead of years or nanoseconds or minutes or whatever. That's as best they can do to see that right now, is those kind of things. These, well, you can see the isotopes stop working when they get at the high temperature, but when they work, they work. Okay, I can make it simple, I can make it complicated. I go with simple. Enzymes are what we're going to be talking about, and they only get produced by bacteria, and they accelerate the rate of reactions by well over 1,000,000-fold. Reactions that would take years in the absence of these enzymes, which are the catalysts, can occur in fractions of a second if the catalyst, which is the enzyme, is there. If you don't have the enzyme, it doesn't happen. You can't wait years to get a chemical reaction in your body. You need it instantaneously. So the bacteria create the enzymes. You don't have the bacteria, you do not get the enzyme. Simple as that. You don't get the chemistry. You see? Moving at the speed of life. This is the American Chemical Society. The speed of life, you need enzymes. Without enzymes, you're done. All right, so don't forget now, it's a systemic disease. That's, I mean, it's throughout your whole body. And the people living with it deserve to have their experiences taken seriously. Now, in a week of extensive test study, patients underwent brain scans, sleep studies, muscle strength, cognitive performance tests, skin, muscle biopsies, blood tests, and gut microbiome and spinal fluid analysis. Gut microbiome. This is where I come in. Now, participants were also put on a controlled diet and spent time in metabolic chambers where their energy and nutrition consumption was measured under stable conditions. So this is a pretty controlled study. And it went on, it was a week, but you get a pretty good idea of what's going on in your body within a week. Now, similar to previous studies, similar. So they said they found a very similar pattern. People with this chronic fatigue had higher resting heart rates. Well, what does that mean? You're not doing anything, but your heart's pumping kind of hard. Well, why would that be? It's trying to make more blood pass through your body to get more nutrition out of that blood, or oxygen, or whatever it's trying to draw from the blood. It's got to keep, and the more you can pass the blood, well, then you're going to get more of it. But you're not producing enough of something. Otherwise, your heart wouldn't be pumping fast. You have a nice, smooth, even, slow heart rate. It means that your body's just sort of having a good time. It's just sort of living life. And everything's consuming correctly, and you're getting all of the nutrition you need, and you can get it in your body easily. So your body just sort of pumps the blood. So they had a higher resting heart rate. Now, which the markers have drawn out an overstimulated immune response that exhausts T cells. And they also had less diverse gut bacteria than the controls. That's the key right there, less diverse gut bacteria. They don't have the enzymes. If they don't have the gut bacteria, they don't have the enzymes. All right, so this group showed no signs of muscular fatigue and performed normally on cognitive tests despite reporting greater cognitive symptoms. You're just tired. You don't have that energy. You're not breaking down the things. You don't have the gut bacteria, is what the studies indicate. And the only way we can know what gut bacteria is the issue is to do some research and to create a database. All right, I probably should leave it at that. But this is, in other words, this has nothing to do with medical advice whatsoever. It's research, and I've been warned by YouTube, to be careful of making any claims. And this is no claim whatsoever. This is just a claim of what the research indicates. So if you can't talk about the research, then I don't know what you can talk about. All I'm saying is this is nothing for you to consider as medical advice, because there is no medical advice here. We don't know. We have no idea. You take a bunch of bacteria, well, it can kill you. Bacteria kills you, but it can also heal you. That's what they didn't realize before. They always considered bacteria, you get bacteria, you want antibiotics, you want to kill that bacteria. Well, that's good. The bad bacteria, and we know there is bad bacteria, no question whatsoever. And you have it in you right now. But you should be able to keep it in check with the good bacteria. So if you don't have the good bacteria, the bad bacteria takes over. It's like in C. difficile. If you look that up, it's a gut infection. It's a terrible, terrible thing, and they can't hardly stop it. So anyway, I'm going to leave it at that, and I hope YouTube will see this as what it is research, trying to understand, but we need involvement, and we are going to need some funding. We're going to need some funding. I've never asked anybody for a penny for anything in my life. But this time, in order to get this done, it's going to have to require some funding, and we have to have some expertise, and we do have that. Andy Marino is the guy. Like I know I talked about him a couple times, and we have a group now that's been investigating this now for a number of years and doing some basically clinical trials. Not like in a clinical study, but just seeing what seems to work and what doesn't seem to work. And all these things have to be brought out now and then really tested. And that's the whole idea of this video, is to let you know that it's bacteria creates enzymes, the enzymes create the chemistry has to be done instantaneously with these enzymes. Otherwise, you get invasion. And you can't fight back. And you can't break down the molecules to support your energy levels. All right, so I'm going to leave it at that. Thank you. I love you all, and I hope we can get some results on this database. And again, it's going to require somebody's going to have to put up some money somewhere, and I don't have it. So So anyway, that's the deal. All right. Thank you.